आयोजित 10 दिवसीय सेंद्रिय शेती उत्पादनातील आधुनिक तंत्रज्ञान प्रशिक्षण कार्यक्रमामध्ये मी डॉक्टर दीपाली कांबळे आपलं मनपूर्वक स्वागत करते. आजचे आपले प्रमुख मार्गदर्शक आहेत डॉक्टर सईद इस्माईल सर, सहयोगी अधिष्ठाता तथा प्राचार्य कृषी महाविद्यालय वसंतराव नाईक मराठवाडा कृषी विद्यापीठ परभणी येथून सर आपल्यासोबत संवाद साधणार आहेत. सर्व प्रशिक्षणार्थींसाठी महत्त्वाच्या सूचना. सर्वांनी आपला माईक आणि व्हिडिओ दोन्हीही बंद ठेवायचे, कारण आपलं हे प्रसारण जे आहे ते युट्यूबवरती लाईव्ह होत असल्या कारणामुळे तुम्ही तुमचा माईक आणि तुमचा व्हिडिओ दोन्हीही बंद ठेवणं बंधनकारक आहे. तुमच्या काही शंका असतील तर तुम्ही चॅटबॉक्स या पर्यायाचा वापर करू शकता किंवा रेज हँड देखील करू शकता. प्रशिक्षण संपल्यानंतर तुमच्या शंकांचे समाधान निश्चितच केलं जाणार आहे. अगदी थोड्याच वेळात आपले प्रमुख मार्गदर्शक आपल्यासोबत जोडले जातील. आजचे प्रमुख मार्गदर्शक आहेत डॉक्टर सईद इस्माईल सर, सहयोगी अधिष्ठाता तथा प्राचार्य कृषी महाविद्यालय परभणी येथून सर आपल्यासोबत संवाद साधणार आहे. नमस्कार सर, आजच्या या कार्यक्रमामध्ये मी पुनश्च एकदा आपलं स्वागत करते. आजचे प्रमुख मार्गदर्शक आहेत डॉक्टर सईद इस्माईल सर, सहयोगी अधिष्ठाता तथा प्राचार्य कृषी महाविद्यालय परभणी येथून सर आपल्यासोबत संवाद साधणार आहे. सरांचा परिचय मी थोडक्यात करून घेते. डॉक्टर सईद इस्माईल सर या अगोदर विभाग प्रमुख मृदा विज्ञान तसंच कृषी रसायनशास्त्र विभाग या पदावरती कार्यरत होते. सद्यस्थितीमध्ये ते सहयोगी अधिष्ठाता तथा प्राचार्य कृषी महाविद्यालय परभणी येथे कार्यरत आहेत. त्यांनी त्यांचे BSc त्याचप्रमाणे MSc आणि PhD चे शिक्षण वसंतराव नाईक मराठवाडा कृषी विद्यापीठ परभणी येथून पूर्ण केलेलं आहे. सरांना ICAR नवी दिल्ली तसेच स्पॉट स्टँडर्डचा आणि मराठवाडा कृषी विद्यापीठ परभणी 2015 सालचा बेस्ट टीचर अवॉर्ड देखील मिळालेला आहे. संशोधनातील उत्कृष्ट कार्यासाठी त्यांना वसंतराव नाईक मराठवाडा कृषी विद्यापीठ परभणी यांच्या मार्फत 2015 साली राधाकृष्ण शांती मल्होत्रा अवॉर्ड देखील मिळाला होता. मृदगंध हा राज्यस्तरीय पुरस्कार 2015 साली सरांना प्राप्त झाला. मराठा महासंघ परभणी तर्फे 2014 साली समाजभूषण हा देखील या पुरस्काराचे देखील सर पुरस्कर्ते आहेत. इंटरनॅशनल क्रॉप रिसर्च इन्स्टिट्यूट अँड सेमी एरिड ट्रॉपिक्स म्हणजे इक्रिसॅट हैदराबाद तर्फे 2009 साली स्पेशल रिकॉग्निशन अवॉर्ड सरांना मिळालेला आहे. त्यांना शिक्षण संशोधन व विस्तार कार्याचा एकूण 27 वर्षाचा अनुभव असून त्यांना शिक्षण संशोधन आणि विस्तार कार्याचा एकूण 27 वर्षांचा अनुभव असून या काळात त्यांनी BSc, MSc तसंच PhDच्या विद्यार्थ्यांना मार्गदर्शन देखील केलेलं आहे. मी देखील सरांची विद्यार्थिनीच आहे. त्यानंतर आतापर्यंत 251 संशोधित अहवाल प्रकाशित झाले असून त्यांनी 160 कॉन्फरन्स मध्ये देखील आपले पेपर सादर केलेले आहेत. यामध्ये राष्ट्रीय पातळीवरील 39, राज्य पातळीवरील 89 आणि 23 लीड पेपर्सचा समावेश आहे. त्यांच्या 11 संशोधन शिफारशी असून सध्या ते सहयोगी अधिष्ठाता तथा प्राचार्य कृषी महाविद्यालय कृषी विद्यापीठ परभणी येथे प्राचार्य पदावरती कार्यरत आहे. सरांचं पुनश्च एकदा मी या कार्यक्रमामध्ये स्वागत करते आणि सरांना अशी विनंती करते की त्यांनी आमच्या प्रशिक्षणार्थींना मार्गदर्शन करावं. सर आज आपल्याला मार्गदर्शन करणार आहेत मायक्रोबियल फॉर्म्युलेशन फॉर सस्टेनेबल एग्रीकल्चर प्रोडक्शन अँड सॉइल हेल्थ. डॉक्टर सय्यद इस्माईल सर, असोसिएट डीन अँड प्रिन्सिपल, कॉलेज ऑफ एग्रीकल्चर, वसंतराव नाईक, मराठवाडा कृषी विद्यापीठ, परभणी. नमस्कार.
Thank you, Deepali. Thank you. I am confused that I should speak in Marathi or I should speak in English because you are introducing in Marathi and I don't know the audience about the audience.
सर सर्व BSc एग्रीकल्चरचे फायनल इयरचे स्टुडंट आहेत, त्याच्यामुळे चालणार आहे तुम्ही इंग्लिश मध्ये.
No problem, no problem. Dear friends and participants, the organizing team, really this is an important topic because when there is a formation of the earth, you might be knowing the first living matter on the earth is a bacteria, first time. And this bacteria, they are having abundance in the soil, in the atmosphere, or in the nature. And they are doing many of the functions. If they stop their function, we will not able to feed ourselves. See, whatever the nutrient they are becoming available in the soil, they are only due to the presence of the microorganisms in the soil. Whatever the digestion is taking place in our stomach, in our large intestine, small intestine, in our large intestine, the microbial flora is there. And that's why the microbial flora is making it easy. Otherwise, you take example of the making of the curd, you take example of the making available nitrogen, available phosphorus, available potassium, available all nutrients, the all nutrient cycles, the microbes are must because they are secreting enzymes, they are secreting many of the material which is responsible for making the nutrients available in the soil itself. And nowadays, such kind of microorganisms are decreasing in the soil due to imbalanced use of chemical fertilizers. I think this training is related with the organics. So I will not talk more on the chemical fertilizers or chemical inputs, but I would like to tell you that chemical use of chemical fertilizers is not bad, or using the pesticide as herbicide is not bad. The imbalanced use or the use where it is not required is bad. The balancing of the nutrients through chemical or through organic is having positive or synergistic effect on the soil health. But the imbalance has a negative effect on the soil health. Soil health means there is a deterioration of physical properties, there is a deterioration of chemical properties, and also there is a deterioration of the biological properties, microbial properties of the soil. And if disturbances in all these properties of the soil, there is no availability of proper or required nutrient to the growing plant. And that's why there is a need of preparation of such kind of microbial bioformulations which can enhance the soil health as well as production of the agricultural commodities. So there is a tremendous pressure on the soil, particularly if there is a pressure on the soil health due to change in climatic condition, natural events. Third is urbanization. Many of the fertile soils, they are coming to the urbanization, construction of the buildings, construction of the industries. And this is also means bad point as far as the soil is concerned. Then agriculture, forestry, waste disposal. All these are the pressures on the soil health. And you see, the human health is dependent on the soil health. If soil health is proper, definitely human health is proper. Because whatever the nutrients in the soil, required nutrients in the soil will be taken up by the plants. And there will be improvement in the plant health. Plant commodities or the food grains, vegetables, fruits, whatever is coming from the plants, humans, they are consuming. If the required quantity of protein, carbohydrates, lipids, vitamins, minerals, if they are coming in the plant system, the human will take it through the foods, and definitely human health will get improved. And if these two things, that is plant and human health, is dependent on the soil ecosystem health. So all ecosystem health is proper, plant health will be proper, human and animal health will be proper. So for the maintenance of that atmospheric balance, animal health, soil microbial community health, and leaching of the groundwater surface runoff, all these things should be managed properly. Then the pressure on the soil health can be minimized. You see, microbiome or microorganisms, they are playing lot of important roles in not only in the agriculture, because in the agriculture, by preparing the bioformulations in the form of biofertilizer or in the form of biopesticide, we are making it proper to utilize most of the nutrients from the atmosphere and the soil. But in the health also, the health people, they are using microorganism for managing the obesities, managing the diseases, and maintaining the nutrition through the microorganisms nowadays. Then environmentalists, those who are working in the environment, they are using these microbes for wastewater treatment or for solid waste treatment, which is coming from the urban areas. And they are also using the environmentalists or those who are working on the soil environmental, soil chemistry, soil environment. They are remediating many of the hazardous chemicals, many of the heavy metals, they are in the soil. And these environmental people, they are remediating, or they are doing the use of microbes. They are using these microbes for soil remediation. Industry people, you take example of the food industry or other allied industries. From the fermentation of the food, very good example, this yeast is used in bakery industries to make your bread and other material. So yeast is used by the food industries. This is a simple example. But many of the microbes, they are used in the food industries, then biochemical industries and biofuel industries. Earlier, I used to work on the biofuels. At that time, we were using the Saccharomyces cerevisiae, this yeast, for the preparation of the bioethanol from the cotton stubbles or cotton stalk. We were using Saccharomyces cerevisiae. So in the coming days, such kind of fuels will also be needed. So in nutshell, I would like to tell you that these microorganisms, they are involved in all the aspects, either agriculture, either health, either environment or industry. Every time they are involved or every place they are needed. I just told you that if microbes are not there on the earth, this earth might have become the heap of garbage. You listen me once more. If microbes were not there on the earth, this earth might have become the heap of garbage, because there is no decomposition, there is no conversion of the organic waste into the nutrients which are required for the plants or material which is required for the animals and humans. So definitely this is playing very much important role in all the requirements of the human beings or in the industry. So what are the beneficial microbes? Beneficial microbes, in another term, we are calling them as a plant growth promoting rhizobacteria, in short term PGPR. Plant growth promoting rhizobacteria are the soil microbes inhabiting around the root surface or in the soil. They are living in the soil or around the root surfaces and are directly or indirectly involved in promoting plant growth and development via production and secretion of various regulatory chemicals. That is called as plant growth promoting rhizobacteria, or nowadays we are also calling them as a plant growth promoting microorganism, PGPM. So these are there in the root rhizosphere. They are there in the root rhizosphere and they are playing direct role, and they are also playing the indirect role, or they are having direct effects. Also, they are having indirect effects. So what are the direct effects? You know, 76 to 78% nitrogen is in the atmosphere. And this nitrogen we are not able to utilize directly. We are not able to take, like we are taking oxygen from the atmosphere through respiration. But human beings and animals, they are not utilizing this nitrogen through respiration. So how this nitrogen, which is present in this tremendous quantity in the atmosphere around us, how it should be utilized for better purpose? So this is being done by microorganisms. In the legumes, rhizobium, or in the non-legumes, azotobacter, azospirillum, and many of the organisms, they are fixing this atmospheric nitrogen and they are supplying to the plant. And this plant is utilizing this nitrogen for the metabolic purposes. It is going to the grains, it is going to the food material, and this nitrogen is getting converted to amino acid, then amino acid is getting converted to the protein molecules, and this atmospheric nitrogen we are utilizing indirectly in the form of protein in our diet. This is nitrogen fixation. Another thing, phosphate solubilization. You might not be knowing, or you people are agricultural student, you are well aware that when we apply phosphatic fertilizers, the efficiency is not more than 20 or hardly many of the times 25 means if farmer is applying 100 kg of phosphate, this efficiency means only 25 kg is utilized by the growing plant. A remaining 75% remains in the soil, and that cannot be utilized as it is unless and until it gets solubilized by the microorganism and it is taken up by the plant. This is the second direct effect. Potassium solubilization. Many of the microorganisms, they are having ability. In Marathwada, in the western Maharashtra, most of the soils are vertisols having clay content more. Vidarbha region, Marathwada region, and western Maharashtra, clay content in the soil is more. And negative charge on the clay particle fixes this potassium, applied potassium in the form of potassic fertilizers. And that's why the efficiency of potassium will also get reduced. So solubilization is also taken up with the help of microorganisms. Many microbial species are there. They are able to solubilize this potassium and to supply the crop. Then siderophore production. In the Maharashtra soils, the deficiency of phosphorus, deficiency of iron, deficiency of zinc, deficiency of nitrogen is more common. These siderophore, they are nothing but chelating agents. And these chelating agents, they combine with the iron and making the iron available to the growing crop. And that's why the iron is taken up by the plant. It is utilized by the human beings in the form of Fe in the diet. It is combining with the protein and forming the hemoglobin in our blood. So these nutrient acquisition is taking place with the help of beneficial rhizopheric macroorganism. Okay, then some of the phytohormone production is taking place. That's why they are called as PGPR. Many of the phytohormones, they are important in growth promotion of the plants. Okay, we'll come to indirect effect of this beneficial microorganism. They are acting as an antifungal, or they are secreting some antifungal metabolites. And these antifungal metabolites, they are responsible for suppressing the fungal diseases in the growing plants or in the soil itself. Many of the microorganisms, they are having induced systemic resistance. They are developing the systemic resistance in plant itself. That's why the attraction of the insect or insect attack or disease attack is reduced. Some microorganisms, they are secreting the exopolysaccharide. Some of the polysaccharide, they are secreted by themselves, and they are protecting the growing plant roots. In the soil, if the exopolysaccharides are secreted, so they are acting as a antifungal agent, antipathogenic agents. Just I told you, siderophore production, ACC diamine production. So they are giving tolerance to abiotic stress, and all these are biocontrol activities. So this is all done by beneficial rhizophoric microorganisms in the soil. You say this is the beauty of soil. Nowadays, the plant, they are grown in the hydroponics, but this beauty of the soil you will not find in the hydroponic. You see, in the hydroponic system, only we are supplying the required nutrients to the growing plants, and we are growing many of the vegetables, many of the flowers in the controlled condition. Okay, here there is no role of any bacteria. But if you see soil itself, and if somebody is growing the crops in the soil, plants in the soil, chemical interaction of metabolites with soil matrix is taking place. Uptake by the microorganism. Many of the microbes, they are responsible for the uptake of the nutrients. Then microbe-derived metabolites, secondary metabolites we are calling, they are secreted. Then organic soil background is formed. This training is on organic. This is not possible in the hydroponics. This is only possible in the soil. Organic cultivation is only possible in the soil. Unless and until soil is there, you are not able to grow any crop or any material in the soil, which is organic one. Root damage is prevented, or root damage is occurring after the completion of the cycle of the plant. And this root, they are going to the soil, they are getting decayed with the help of microorganisms. And extraction from the soil, this extraction of the nutrients is taken place. And these material nutrients, they are coming to the food chain, and we are utilizing them. So what the PGPRs are, basis of location. They are either extracellular or they may be intracellular. You see, these organisms might be outside of the cell, or they may enter in the cell itself, and they are acting in both the way. Many of the microorganisms, they are entering in the cell, like in the sugarcane. We are giving gluconacetobacter. It is intracellular organism. It is entering in the plant, and it is fixing atmospheric nitrogen. But many of the microbes, they are extracellular and working in the soil, are working in the root rhizosphere. Root rhizosphere, what is that? I will show you. Basis of function, what kind of plant function they are performing. Plant growth promoting bacteria. They are involved in the promotion of the plant growth. They are controlling the bacteria, or they are controlling the diseases which are occurring through bacteria, called as a biocontrolling bacteria. Then stress humoregulating bacteria. Many of the stress is there. That is the heat tolerance, that is the salt tolerance, that is the salinity tolerance. Such kind of tolerance mechanism is being performed by the plant growth promoting rhizobacteria. Then PGPR on the basis of activities. Which activities they perform? They are acting as a biofertilizers. They are acting as a phytostimulants. They are stimulating the phytohormones. Then rhizoremediators. I told you, remediation means the destruction of the heavy metals. Example, destruction of the toxic metals or toxic material. It is being also done by the PGPR. Then they are acting as a biopesticides. They are protecting the plant from fungal diseases. They are protecting the plant from bacterial disease or all the pathogens. And they are also acting as a bioprotectants. They are protecting biologically, not only the growing plant, but they are also protecting the soil. What are the desirable traits? Good traits should be considered when you are preparing any bioformulation, microbial bioformulation. Number one is microbes for atmospheric nitrogen fixation. Either it should fix atmospheric nitrogen in large quantity. Microbes for phosphorus solubilization, these are also available with us. Microbes for potash mobilization, these are also with us. Microbes for micronutrient solubilization, these are also with us. So these are some desirable traits, desirable characters of the microorganism. And these characters should be considered when some microbial formulation is to be prepared and is given to the farmer. Then microbes for plant growth regulators, microbes for bioremediation, microbes for salinity tolerance, microbes for drought stress tolerance, microbes as a biocontrol agent. So all these are the desirable characteristics of the microorganism. Some may have 1, 2, 3, some may have one of them, like rhizobium, it can fix the atmospheric nitrogen. Azotobacter fix the atmospheric nitrogen. Azospirillum, it is fixed atmospheric nitrogen. Itna hi humko pata hai. But that particular microorganism, they are also having other desirable traits which are needed to be studied. For example, if Rhizobia azotobacter azospirillum, in addition to fixing the atmospheric nitrogen, they are also performing the other functions which I have listed here. They can act as a plant growth regulator also. Many of the time they are giving salinity tolerance. Many of the time rhizobium is also giving drought tolerance. Many of the time the rhizobium azotobacter, PSB, they are also acting as a biocontrol agents. So many of the functions are performed by one organism, or some organisms can also have different abilities or different traits which are desirable traits. And these are the PGPR. So what is rhizosphere? Where the abundance of the microbes are. It is the zone of soil that is significantly influenced by living roots. Where the living roots are there, usually extend about 2 millimeters out from the root surface. This is the reach in, particularly, this is the reach in organic material due to the secretion of root exudates. And here, microbial population or microbial activity, enzymatic activity is 2 to 10 times greater than the bulk soil. So this is the living zone where the microbes are there, where enzymes are there, and many of the activities are going on simultaneously. Mechanism of plant growth promotion, how it is doing. Beneficial microbe facilitate plant growth directly by either assisting in resource acquisition. I told you, they may fix atmospheric nitrogen, they may produce hormones, they may help in the nodulation. In the legumes, nodulation is important. And this nodulation is helping. More the nodulation, more the living nodules, more the atmospheric nitrogen fixation. They are also helping in nutrient uptake. All the nutrients. Now we are not talking about the PSB or not. We are talking about the nitrogen fixation. We are talking about the nutrient uptake by dissolving or making all the micronutrient secondary nutrient available. Siderophore protection. Siderophore is the chelating agent, which is having ability to combine with the metal ions. And these metal ions, which are non-available for the crop, they are becoming available and they are taken up by the plant. Many of the microbes like trichoderma, this is a fungi. Many of the species of the trichoderma are there. Trichoderma viridi, trichoderma harzianum, and many other trichoderma species, they are acting very good biocontrol agent. You might be knowing our university is selling the biomix to the farm. And the turnover or sale of the biomix this year up to February, today, it is 3.5 crores. Not talking about the lakhs. University sold biomix to the farmers for biocontrol or for making the nutrient available, and earn 3.5 crores with the sale of this biomix, which is a very good biocontrol agent and very much demand by the farm, which is this biomix is nothing but microbial bioformulation. We have given the name biomix. So such kind of bio or microbial formulations we are selling in the form of biofertilizers also. This slide I have given. So what the PGPR they are doing, one by one we check. This PGPR is secreting ACC-D-aminase, cytokinase, gibberellins. All these are the plant promoting agents, and their growth augmentation is taking place. Metal solubilization, zinc is a metal, then iron, manganese, copper. These are the essential metal ions. They are becoming soluble through microorganisms, and they are also increasing the growth of the plant. Many of the enzymes like nitrogenase activity, antifungal activity, and many of the antioxidants, they are secreted by the plant, which are also helping the plant growth. The microorganism, they are also secreting the antibiotics. Antibiotics means these antibiotics is affecting on the pathogenic bacteria, and they control the phytopathogens. Enhanced nutrient uptake is also with the help of only these microbes, PGPR. So in the direct mechanism, I will not go in detail, time will not permit me, but you remember nitrogen fixation is most important one, which is called as a biological nitrogen fixation. And this is being done by the rhizobia as well as some of the organisms which are doing in the soil itself. And we are utilizing the atmospheric nitrogen indirectly in our food in the form of protein. So this is the direct mechanism being done by microorganisms. Then phosphorus. Phosphorus is becoming soluble with the help of microorganisms. You know, phosphorus we are adding to the soil through mineral fertilizers like single superphosphate, diammonium phosphate. Some of the surface minerals are there, and some primary minerals, some secondary minerals at the time of feathering. And what happens? The phosphorus in the soil is fixed in the form of calcium phosphate in the alkaline or vertisols, or where clay content is more, where calcium content is more. But in the acidic soils, the alumina and Fe is more. The phosphorus which is added through external source, it is getting converted to alumina phosphate and iron phosphate and becomes unavailable. At that time, there is a role of microorganism or PSB, phosphate solubilizing bacteria. They are converting this unavailable phosphorus and giving to the plant. And this phosphorus coming to the soil solution and becoming available in the form of HPO4 or H2PO4. Many of the time, some quantity of this phosphorus is leached out, a large quantity is taken up by the plant, and the crop is harvested with phosphorus, which is going to become a food material. Then how it is added? Animal death and decay. Many of the organic form of phosphorus, they are there, and they are also mineralized simultaneously with the help of microorganisms. This is also same. How they are doing? The microorganism, they are secreting some of the acids. They are called as organic acids. And these organic dilute acids, they are responsible for converting this unavailable phosphorus, that is calcium phosphate, aluminum phosphate, or iron phosphate, and it become available to the growing crop. Acid phosphatase and alkaline phosphatase, some of the exopolysaccharide phytases, they are responsible for making the phosphorus available to the growing plant. So what microbial formulations, they are nothing but biofertilizers, biocontrolling agents. They are called as a microbial formulations. So these are biofertilizers. They are defined as a product which contains one or more living organisms. Living organisms. When applied to the soil, seed, or surfaces of the plant, colonize the rhizosphere of the plant internal tissues, and they make the nutrients available, or they act as a plant growth promoting organisms. So I will say step by step why the farmer is interested to utilize or to use the biofertilizers. Their characteristics, ability to convert the unavailable form of the nutrient to the available. This is one thing. They improve soil properties, particularly the microbial properties of the soil are getting improved with the help of biofertilizers or microbial agents. Then use of biofertilizer not only make atmospheric nitrogen available, but also make the phosphorus, potassium, and micronutrient available also. They also enhance the release of hormones in the soil, vitamins and oxygens in the soil. They also help to proliferate beneficial microorganisms. Synergistic effect. One organism is helping to the another organism to proliferate in the soil. That is called as a synergistic effect, helping to reduce deficit in fertilizer consumption. So if inorganic fertilizers are not sufficient to add in the soil, these biofertilizers, they reduce the cost on fertilizers, and they also cheap and easily available. The biofertilizers, they are cheap in the market. They are available in all the universities. See the scenario. This is the very recent data published in 2021 research paper. See, biofertilizers and biocontrol agents. If you see, 50% are used by the farmers. 50% total quantity what is available in the market. 50% biofertilizers, 50% biocontrol agents. And many of the farmers, they are using both bacteria and fungi. Large quantity of fungal biocontrol agents are available in the market. And many of the farmers, they are using bacteria as well as fungi, both. Okay. And you see PGPR, large quantity of PGPR is used in the agriculture, followed by mix, epiphytic fungi, soil fungi, and some are rhizosphere. Very few quantity of rhizosphere is used. Mycorrhiza is also used throughout what if you see the data. Large quantity of mycorrhiza, which is called as AM fungi, arbuscular mycorrhiza, is used and which is available in the market also. Come to the liquid biofertilizer and consortia. You remember, the biofertilizer itself is a mix, itself is a living material, living microbes are there. But if I say consortia, it is a mixture of one or two organisms. So in earlier days, we used to give this in the form of powder, but there is a sowing of the seed with the help of seed drills, and this seed drill is getting choked up due to the material, powder-like material, which is used as a carrier material. So we have converted, or we have used some material which is a cell protectant, and we have converted this or life of these microorganisms which are giving to the farmers for a long period. They are called as a liquid biofertilizers. Or liquid biofertilizer may be the rhizosphere, it may be PSB, it may be KSB, or it is zinc solubilizer, whatever it may be. Or many times it is a mixture of N fixers and P solubilizer. That is called as our university is selling in the name of rhizophos, or we are selling it as azotophos, or we are also selling in our university through NPK biofertilizers. And there is a large demand. Why liquid biofertilizers, or why consortia should be used? That is a mixture of two or more organisms is called as consortia. That is to stabilize the organism during production, distribution, and storage. Easy is there. Liquid biofertilizer, they are easy to produce, easy to distribute to the farmer, and easy to store in the houses of the farmer. Easily deliver to easily deliver in the field in the most appropriate manner. The biofertilizer can be used as a seed treatment. It can be applied through drip irrigation. The biofertilizer can also be used for seedling dipping. If vegetable is there, if vegetable is sown already in the field at that time, it can be drenched through the knapsack sprayer in the soil itself to protect the microorganism from harmful environmental factor at the target site, thereby increasing persistence for longer time, and to enhance activity of the organism at the target site by increasing its activity. When it is going to the soil, its activity should be for longer time. So the liquid biofertilizers are better as compared to carrier-based biofertilizers. Some of the results I will show. I will not show all the tables, but some of the results I will show with the help of table. So yield of various crops in inoculated and uninoculated field trials. Take example, soybean we have taken. In inoculated, this much is the yield, and in inoculated, the difference is inoculation of rhizosphere or PSP, 23% increase in the yield. In case of safflower, 17% increase. In the sorghum, we find 41%. In the chickpea, 24%. In the cotton, 22%. Many of the farmers, they are getting surprised that this is a minute input, which requires only the timely application, and it is having tremendous effect on the yield of the crops as well as improvement in the soil. So we have also conducted some frontline demonstration on the farmers field, and we found on the farmer field also there is an increase in the yield up to 27% with the application of rhizosphere and PSB, and in the groundnut 23% increase in the yield is there due to only inoculation of the biofertilizers, microbial biofertilizers. So these are some of the values. We have conducted the experiment for 3 years, and you see the value. This Bradyrhizobium japonicum and this is Bacillus megaterium, which is a PSB. You see the result. The increase in the yield is there as compared to the one inoculation. If you compare only with recommended dose of fertilizer, this is a recommended dose of fertilizers yield, and this is with fertilizer and with microbial. 5 quintal increase in the yield is there. Similarly, this is the yield of wheat. Here also the tremendous increase is there. If you compare with only chemical fertilizers, near about 7 quintal increase in the yield with only biofertilizers when you compare with recommended dose. Improvement in the soil property. Most important for us is organic carbon content. You see in the uninoculated control, the carbon content is very low. But when both the organisms were inoculated and we grown the wheat, there is an increase in the organic carbon content in the soil, which is important for the microorganisms and to improve the soil properties. Same is the case with the availability of nutrients in the soil. If microorganisms are used in addition to recommended dose of fertilizers, there is a more available nutrient after harvest of the crop. And these nutrients can be utilized for upcoming crop, next crop. There is many of the times I used to say, or I used to speak that the soil, it is a living material. And when bacteria, actinomycetes, fungi, they are more in the soil, microbes, they are more in the soil, definitely it will be living. Otherwise, if imbalanced chemical fertilizer they are applied, imbalanced herbicide application is there, imbalanced pesticide application is there, the soil will be dead, and dead soil cannot convert the unavailable nutrient to available form. So tremendous quantity of microbes will be there if these were inoculated for the seed or they were applied through drip irrigation. Advantages of some liquid bioformulation of biofertilizers or consortium, high population can be maintained. You remember 1 ml of the biofertilizer, 1 ml of microbial formulation contain 10 raised to the power 8 cells. And it can be stored for 12 to 24 months. If farmer is taking it, he is able to store it for 12 to 24 months at room temperature. Then less chances of contamination in the liquid biofertilizer. Chances of contamination is not there because bottle is packed. No effect of high temperature. 45 degrees centigrade temperature is there in the summer season. There will be no adverse effect. Then cost saving on carrier material, definitely, because carrier materials should be purchased from either from Gujarat or from Tamil Nadu. So the lignite or the talcum powder is a carrier material used. Here in the biofertilizer, liquid biofertilizer, no carrier material is required. And quality control is easy. Better survival on the seed. And this can be used for drip irrigation also. The dose of the biofertilizer through drip is 1 liter per acre. The 1 liter we are selling at the rate of 375 rupees. So required quantity is 1 liter and required amount is 375 rupees, and very good results in case of drip irrigation to the fruit crops or to the, in our area, many of the farmers, they are having drip to the cotton, drip to the sugarcane, drip to the banana, and they are using it. Why consortia? You remember the consortia I told you, microbial consortia is a mixture of 2 or more interacting microorganisms. And this microbial consortium or microbial bioformulation, it can perform many of the functions at the same time. That's why consortia should be used instead of single microbial biofertilizers if it is available. If it is not available in the market, then one inoculation after second inoculation is also possible to the seed or through drip irrigation. So consortia is better as compared to the single microbe biofertilizers. Then nowadays you might be knowing biocapsules, which is the most advanced, and we are calling it as an inoculant for the future. You know, Indian Institute of Spice Research is there in Kerala, and they have developed one biofertilizer, or they have developed the technology for the biofertilizer preparation, which is in the form of capsule. They are selling at the rate of rupees 100 per capsule. What they are doing in the 100 to 200 liters of the water, if one capsule is dissolved, that makes the whole 100, 200 liter of the water as a biofertilizer. And that water can be applied through drip irrigation, or it can be used for the treatment of the seedlings of the vegetables. It can be used in any form. They contain all the microorganisms which are there in the capsule itself. So this is a inoculant, or this is a microbial formulation in the coming days all over India. I will not go in detail how to prepare steps to be followed for in potential microbial consortia development. I just told you, thoda sa leta hu, so that those who are going to prepare the biofertilizers in the coming days, those who want to become the entrepreneur, they will be able to do it. Screening and characterization of the PGPR. First of all, you should see which organism is there, which is having very good traits or very good ability to fix atmospheric nitrogen, solubilize phosphorus, or they act as a PGPR. Then assessing multifunctional PGP. If plant growth promoting activity is more in case of bacteria, that should be selected. Then their characterization should be done with the help of molecular characterization technique, which is uska naam pata hona chahiye. So the characterization should be done. It's complementarity test. If you are preparing the consortia, making the consortia for one or two organisms, whether it is compatible with each other or not, that should be tested. Then biocontrol potential. Is it able to protect the fungal diseases, bacterial diseases? That also should be tested. Then directly we are preparing the consortia with the help of these organisms, and they can be evaluated in the field itself. So microorganisms, they are having very good abilities. These three I have listed here. Mutualism. Microorganism, they use mutualism. One is helping to another, another is helping to other organism in the broth or in the laboratory, and that is good. Then commensalism. They are having the commensalism means they are fungi and bacteria. If they are mixed in and it is given to the farmer, they are acting as a facultative and applicate herbs can be mixed, and they commensalize each other. Then protocooperation, one is cooperating to the another organism where they are going in the soil. So such kind of consortia or bioformulation can be prepared and used to enhance the growth of the crop and to enhance the soil health also. So how these microbial consortia or microbial bioformulations can be prepared? Here possibilities are there. Synergistic beneficial effects. When one microorganism should enhance the activity of other organism, that is called as the synergistic effect. So when PGP, one organism, bacteria with bacteria, growth stimulation, nutrient uptake, group development, stress tolerance, all these abilities should be there. These microorganisms should be selected for preparation of bioformulation or consortia. Many of the time, AIM fungi can be mixed with the PGPB, that is the plant growth promoting bacteria, and can be made a bioformulation. Trichoderma can also be mixed with the PGPB if cell protectant is used. If cell protectant is used, trichoderma can also be used with any bacteria, like we are doing in the biomix. And this material can be utilized to apply to the seed, to apply to the plant, growing plant through drip irrigation or through any other method, so that it will go to the rhizosphere, it will multiply, and it will give plant growth, and it will give fitness to the growing plant. I will not explain formulation of EM, effective microorganisms. These are the detailed slides, but you see, formulation of microbial consortia or microbial formulations at ICR ANP on soil biodiversity and biofertilizer. One unit is there, which is given by ICR BNMK Parmani. Here we are preparing the microbial inoculants with multiple organisms. Development of new inoculant formulation with multifunctional microbes. They are functioning. Many of the microbes we are adding, and they are functioning in a different way. They are acting as a nitrogen fixers. They are acting as a phosphate solubilizer. They are acting as a potash solubilizer. They are also acting as a antipathogens. They are also acting as a antifungal agents. They are called as a microbial inoculants, mixture of the microbial inoculants, or they are also called as the microbial bioformulation. In this microbial consortium, greater importance is given to the sustainable agriculture. That consortia should enhance the yield as well as it should enhance the soil health. Then steps are involved. Large production of the biofertilizer or bioformulation is possible with the help of some steps are given. This material is required if we are going to prepare the azotobacter broth, then rhizobia, then for phosphate solubilizing bacteria. First of all, there is a preparation of mother culture. And I told you that how mother culture should be selected. If bacteria is in good traits, they should be selected and their mother culture should be prepared. Then after that, prepare a broth in the laboratory. Then preparation of liquid biofertilizer by cell protectant. This is important. Liquid broth, 40-liter broth for the preparation of 100 liter of the microbial bioformulation. We are taking 40 liters of the liquid broth. Then we are adding the cell protectants, 25 liters sterile water, only 75 gram trehalose, then 100 gram arabinos. This is FeEDTA. These are acting as a cell protectant. I told you that these liquid biofertilizers, liquid microbial consortia or liquid microbial bioprotectant can be stored for at least 1 year. Their activity do not reduce. 1 ml can contain at least 10 to the power 7 microbe for 1 year. And for that purpose, these cell protectants are added. This is the list I have given. Then filling and packaging of the bottles. Just after preparation of the liquid biofertilizer, as mentioned above, they should be filled in the plastic bottle. Automatic is there as well as manual filling is there. And on the bottle, name of the manufacturer, license number, name of the inoculant, all the details should be written. Many of the Indian companies or many of the industries, they are preparing the consortia bioformulation. So these are the names of the Indian industries. They are selling in the Indian market and they are having very good effect on the production as well as they are having very good effect on the soil health. The conclusion is this: microbial inocula, either single or consortia, have many advantages than limitations. They can restore soil fertility, improve and enhance nutrient availability. They protect against biotic and abiotic stresses. They increase soil microbial activity, decomposes toxic substances like heavy metals are there. Then promote colonization of mycorrhiza and other useful microbes, help in recycling of organic matter. Microbes, they are having ability to decompose the organic matter also and increase plant defense. They also increase the immunity for suppressing unwanted parasitic and pathogenic microorganisms or attacks. Each year, there is a nearly 12% increase in the demand of microbial bioformulation or inoculants. Each year, the demand is increasing by the farmers. PGPRs such as azotobacter, bacillus, azospirillum, many of the names are there, are now being commercially produced at a large scale. Many of the, I have shown the names of the industry they are producing and selling in the market. All the universities in Maharashtra, 4 universities in the Maharashtra, they are also selling the biofertilizer to the farmers in a large quantity. Studies have reported microbial consortia containing all the desired traits need to be explored in all the crops. There is a need to use these microbial inoculants, microbial bioformulation in all the crops. Either it may be legume, it may be cotton, it may be sugarcane, or it may be any commercial crop, it may be fruit crop, they may be vegetables. In all cases, the students, they should promote the farmers to use this microbial bioformulation or microbial fertilizers or biofertilizers of consortia so that there will be enhancement in the yield sustainably, as well as there will be enhancement in the soil health. Thank you.
Thank you so much, sir. कोणाला काही प्रश्न असतील, if you have any query, any question regarding today's, then you ask directly to the sir. Otherwise, you can type in a chat box.
If somebody want to ask, please raise your hand so that you will be unmuted, or unmute is needed. You raise your hand. If no
कोणाला काही
question,
प्रश्न असतील, काही समजलं नाही असं असेल तर तुम्ही तुमचे प्रश्न विचारू शकता.
but
Please
how, but
unmute
how,
yourself.
but how,
Please
madam, they
unmute
will,
yourself.
or put your question in the chat box.
Yes.
If no question, I thank you all. If no question, I thank all, and I think
Thank you, sir. On behalf of all my KVK staff, Krishi Vigyan Kendra Badnapur, Jalna II, I extend really heartily a vote of thanks to our today's expert and guest also, Dr. Said Ismail sir, Associate Dean and Principal, College of Agriculture, Vasantara Naik Marathwada Krishi Vidyapith, Parbhani, who spared time for from his very busy schedule to grace this event and also guide us. So thanks a lot, sir. पुनश्च एकदा तुमचं मनपूर्वक आभार सर. तुम्ही आमच्या सोबत जोडले गेलेत आणि खूप छान मार्गदर्शन केलं. सर एक रिक्वेस्ट आहे तुम्हाला की तुमची ही जी पीपीटी आहे वर्ड फॉर्मॅट PDF फॉर्मॅट मध्ये जर आमच्या सोबत शेअर केली तर खूप छान होईल. आणि कोणाचे काही प्रश्न असतील तर कृपया विचारा.
immediately I will share on your email, or I should share on WhatsApp.
Email, WhatsApp, whatever.
Okay, I will share.
Okay. Thank you once again. Thanks to all the people who have directly and indirectly helped to make this event successful. Once again, thank you.